In previous lecture, we have already discussed about eukaryotic cell and the organelles of eukaryotic cell. In unit number 8, the cell, the unit of life. In a previous lecture, we discussing about endomembrane system, in which we already discussed about endoplasmic reticulums. And in the endomembrane system, there are 4 organelles. First one is endoplasmic reticulum, another one is a Golgi body, third one is a lysosome, and the fourth one is vacuoles. So why we call these 4 organelles as a endomembrane system? Because these all organelles is function in a coordination. That's the only reason why this double membrane 4 organelles considered as a endomembrane system. Today we will discuss about another endomembrane system organelles and another organelles of eukaryotic cell. So let us start with the second endomembrane system organelles, that is Golgi apparatus. Golgi bodies or Golgi apparatus, which is discovered by Camillo Golgi, which is found in a cytoplasm, and it is a densely stained organelles which is found in cytoplasm. Golgi body is a reticular structure found in cytoplasm. This is the figure of Golgi bodies. It consists flat disc-shaped sac which is arranged in a parallel manner. You can see here, these are the sac, or also called as cisternae, which is arranged in a parallel manner and form 2 regions. First one is a convex region, also known as a cis region, and another region is a trans region, which is also known as in a concave manner. So the convex cis region is found side by nucleus, where concave region found opposite side the nucleus. The function of Golgi bodies is to packaging the materials. How these materials are packaged in Golgi bodies? So the material is to be packaged will come from the endoplasmic here nucleus, and it is a endoreticulum system. The material which to be packaged is come from endoplasmic reticulum. Here it is known as vesicles, which enter in the Golgi bodies from cis region, and then it transfer to the trans region, in which these vesicles are modified and go up from the trans region and come out from the trans region. The another function of Golgi bodies is to modify proteins. Golgi bodies प्रोटीन को मॉडिफाई करते हैं कैसे? तो एंडोप्लाज्मिक रेटिकुलम के ऊपर जो राइबोसोम्स होते हैं वो हम सब जानते हैं कि राइबोसोम्स इज factories of protein synthesis, factories for protein synthesis. The proteins which synthesized from ribosome will enter in the cis region and come out from the trans region, in which the proteins modified into particular manner or particular requirement. So this is the about Golgi bodies. Now another organelle is a lysosome. So what is lysosome? Lysosome is also double membrane structure found in the cytoplasm. When the vesicles of Golgi bodies join with the solid particle which enter in the cell from the plasma membrane, here from one solid particle enter in the cell. So what will happen? The vesicles from Golgi bodies cover the particle and form lysosome. This structure known as a lysosome, which is double membrane structure. and having hydrolytic enzymes, which means it hydrolyzes the molecules like proteins, lipids and carbohydrates, and it works in a acidic medium. Sometimes this lysosome also digests or hydrolyzes the own organelles, which means जब वो स्टार्वेशन में अपन लोग होते हैं तो वो अपने ही ऑर्गेनल्स को डाइजेस्ट कर लेता है, उसी की वजह से लाइसोसोम आल्सो नोन एज अ suicidal bag of the cell. This can be asked in MCQ that which organism is also known as a suicidal bag of cell. So lysosome is considered as a suicidal bag of the cell. Now another organelle is a vacuoles. As we all know that vacuoles is only present in plant cell. On other hand, in an animal cell, vacuoles is absent. If you direct your attention over here, then you will realize that in plant cell, maximum area is covered by one empty space known as a vacuoles, which is covered by only one layer, only single layer known as a tonoplast. This tonoplast facilitate the active transport of certain molecules against the concentration gradient of that particular molecules. Now another organelle is mitochondria. Mitochondria is one of the most important organelles in terms of respiration and formation of energies for the cell. The main function of mitochondria is cellular formation of cellular energy for the cell. Now let's see the structure of mitochondria. This is the outer membrane of mitochondria, and then this is the inner membrane. The inner membrane is folded inside, known as cristae, and the inside the inner membrane matrix is present. The matrix is filled with the homogeneous substance, and in matrix ribosomes and DNA of the mitochondria also present. Now this is the structure of mitochondria. Let's see the ultrastructure and their function of the mitochondria. The diameter of mitochondria is 0.2 micrometer to 1 micrometer, and the length of the mitochondria is 1 micron to 4.1 micrometer, which is varied in nature in a different cell, and the basis of their physiology and their function. The inner membrane is provide the larger surface area for the aerobic respiration in all the organisms, and the mitochondria is specific site for the aerobic respiration in an organism, which is responsible for formation of ATPs or cellular energy, also known as the ATP. ATP, the full form of ATP is adenosine triphosphate, which is the one form of energy. When this adenosine triphosphate break, it will produce a certain amount of energy, which is useful in the function of cell. This DNA also have ribosomes and their DNA. The ribosomes which is present in the mitochondria is 70S, which is similar to the prokaryotic cell or bacterial cell, and DNA is also present. This mitochondria, one interesting thing is that this mitochondria is not produced by our body, it is divide and reproduce themselves by the binary fission. Consider this is the mitochondria. This will, like bacteria, this will divide into two cells. So some scientists say that this is not our organelles. This is a mitochondria is a organelles, which is maybe it is a bacteria which comes in our cell or comes in our body by the endosymbiotic system. So this is all about mitochondria. Now we will discuss about plastids. So next is plastid. We all know one kind of plastid is chloroplast, but when we see different kinds of plant and different kinds of leaf of plants, we confuse that why these plants have different shades of colors of green plant, because there are three different kinds of plastid which is responsible for different color in the leaf. For example, plastid number 1 is chloroplast, chromoplast and the leucoplast. The chloroplast, first one is a chloroplast, which composed of chlorophyll. In chloroplast, one pigment is present known as a chlorophyll, and another pigment is a carotene, which is responsible for green color in plants. The another plastid is chromoplast. The chromoplast contain carotene and xanthophyll. This chromoplast is responsible for production of different colors like yellow and light green and red color and another. So this chromoplast plastid is responsible for different shades of leaf, different shades of green color in a leaf. And the third plastid is leucoplast. The leucoplast is a colorless plastid which is present in a plant, and the function of this leucoplast is to store a particular different kinds of food. For example, it store carbohydrate in an amyloplast and protein and lipids. So this if question comes in an exam that which plastid is responsible for storing the food in the cell, so the answer is that leucoplast. Now let's see the ultrastructure of chloroplast. As we all know that chloroplast is responsible for the production of sugar, which is also known as the food of plant, by the process called photosynthesis. The chloroplast is very abundant in a type of cell known as mesophyll cell. The chloroplast is lens-shaped and discoid in shape, which means the chloroplast in plant which is present in a different shape. For example, one shape is a lens shape and another shape is a discoid shape. And the chloroplast in plants present in a different number also. For example, in a chlamydomonas, it is present in one chloroplast in a one cell, where in a plants and another higher organism it is present in a 40 to 50 per chloroplast per cell. Let us see the ultra structure of chloroplast. The chloroplast is composed of double membrane, the outer membrane and the inner membrane. Inside the inner membrane, the space limited by this inner membrane is known as stroma. Stroma is site for the production of sugar in the plant, and in stroma, the plated membranous sac-like structure present known as thylakoid. So this sac-like structure found in stroma is known as thylakoid, and the membrane, the space of thylakoid membrane is known as lumen, and the stack-like structure of this thylakoid arranged in a piles of coin and from a structure known as a grana or granum. It is known as a grana. Like mitochondria, this chloroplast also have their DNA and their own RNA. The RNA is in a chloroplast is 70S. Here S is a Sweetberg unit, which stands for the rate of movement in a centrifugation of these molecules. So this is about chloroplast. The function of chloroplast is to production of sugar for the plants. So this is about plastids. In next video we will discuss about flagella and then discuss about nucleus, which is the last topic of our unit name is cell, the unit of life. So thank you.